So a long time ago, I heard the story about the evolution of the eye, but I really liked it because it built up the structures of the eye through evolutionary processes in a similar way that I imagine we might construct an eye conceptually. So I was wondering first, do we actually have that story? Because it's a really like complex organ, your eye, right? There's a lot of parts that all seem to fit together.
So I guess there's a couple different ways of approaching this question. One is to look at the kinds of eyes we have in extant species, so species that are living today. And this is an approach that evolutionary biologists take when we're trying to understand the evolution of a trait, like a complex trait like an eye. We look across species and look at features of a trait that are shared between species and those that are that differ. And so if we look at living organisms, we can see everything from organisms with just a single neuron that's light sensitive. And from there, we can look at other organisms that have slightly more complex organization. So maybe they have an array of these light sensitive neurons together with an array of pigment cells. Those can be organized into like sheets, or they can be organized into cups. If you have light sensitive cells organized into a cup, the advantage of that is that you begin to be able to pick up directionality of visual scenes. And the downside of that is that a cup isn't very good at focusing an image. However, we do see other organisms that have started to solve that problem. So that cup might end up with a small pinhole opening. And so that pinhole actually helps with this problem of focusing light. And then in other organisms, we might see, in addition to the pinhole, we start to see what looks like a lens, which even more helps with focusing the light, and so on and so forth. And in fact, a lot of these examples of eyes are found in marine organisms, and the ones that have sort of the pinhole type eye, actually let in water from the ambient environment as into their eye. And then you do see then other organisms where that chamber gets closed off and you have an actual lens. So in extant organisms, we can see very, very simple eyes from a light sensitive neuron and a pigment cell to very complex eyes that very much look like ours. These would be the eyes of things like an octopus. And then, so if you're an evolutionary biologist, what you would then do is you would want to know how old are these groups, how back in evolutionary time did these various organisms evolve? And then you begin to infer like when each of these stages of eye development occurred. And so that is the approach that people in my field would take to it. If I were a paleontologist, if I were studying fossils, I have this additional approach, and they actually are complementary, and that is that we can see in the fossil record that about 538 million years ago, when there was a period known as the Cambrian explosion, we see almost instantaneously the appearance of two major types of eyes. Before that period in the fossil record, we really don't have fossils that we can recognizably discern as containing eyes. But around 538 million years ago, all of a sudden we see organisms where we have these kinds of eyes, and the eyes that we see represent the kind of the two major branches that we think about today, which is the vertebrate type eye, and then what we call the invertebrate type eye that we see, for example, like in insects.
So I work with optics, and we have to focus images and things like that. And when I teach people, I start with the pinhole, because the first cameras, if you want a photosensitive sheet of paper, you can't just hold it up and get a picture, right? Because you're getting light from everywhere. So if you want to focus light from a specific place onto the specific part of your sensor, first way they did is just to put a pinhole, right? But that's not very good because you don't get a lot of light that way. And so you have a lens then, which focuses all the light that hits the lens into a spot. Yeah, so that kind of follows the evolution of animals, right? They start off with just this light detector. You point in a direction, okay, the light's that way, that's useful to me. And then they say, okay, it's actually useful to try and build an image then. And so yeah, I really like that story. So can we do a little bit more into the structure? I want to go into like all the different parts of the eye and then compare it to the butterflies as well. I guess maybe the first part would be those photosensors, which I know is what you study a lot.
Yeah, so we spent a lot of time studying these photoreceptor molecules that have different names, but they're basically composed of two parts. One is a protein. It's known as an opsin protein. It's part of a very large superfamily of proteins, many of which are involved in different senses like olfaction, taste, vision. And that protein is bound to another molecule that is the molecule that is actually UV absorbing. So by itself, it has a UV absorbance, but when it's bound to the opsin protein, it has a series of electrons that their position in the molecule is affected by what amino acids are in proximity to that light sensitive molecule. And so you can take the chromophore, which by itself absorbs in the ultraviolet, and together with an opsin with a particular amino acid sequence, you can get a molecule that's either sensitive to more shorter wavelength UV light or longer wavelength light, so blue, green, and red. So simply by changing the amino acids of the protein, you can tune the chromophore to be able to absorb shorter or longer wavelength light. And so when it absorbs a photon of light, that chromophore undergoes a conformational change. That conformational change is translated to the opsin protein, and the opsin protein sets off a signaling cascade in the photoreceptor neuron that eventually gets transmitted, if the animal has a brain, to the brain, that there's light that's been detected.
So that's an electrical response, or
It's
is
an
it a
electrical
physical?
response. No, it's
Okay.
definitely electrical response. Yeah.
Okay.
Although these rhodopsin proteins have also been shown to actually have a mechanical property to them because they're found in membranes, and they actually can pull on the membranes. But that's sort of work that's people are just really beginning to unravel.
When we learn about this most basic parts of vision, right? You have rods and cones, and so these proteins make up the cones. Is that right? Okay.
Yeah, so there's a rod opsin, and there's in humans there's three types of cone opsins. There's a blue, a green, and a red. And those proteins can shift the absorbance spectrum of the visual pigment or photopigment to in the case of humans, the peak is in the blue, the green, or the red. Yeah, so for human vision, we think about our color vision mostly being determined by input from those three pigments, the blue, the green, and the red. The reason why we need more than one is that a single pigment is actually sensitive to, if you can imagine, like a bell-shaped spread of wavelengths. And so if you only have one pigment, you might be able to detect a photon within a particular range, which is that, under that bell-shaped curve, but you can't tell the wavelength. So you might be able to tell that you've detected a photon, but you don't know what the wavelength is. You need two pigments with different absorbance properties that are, in most cases, overlapping in order to be able to figure out what the wavelength is. And that is what allows us to have color vision, is having more than one kind of photoreceptor cell that's sensitive to different wavelengths of light.
Yeah, the way it helped me conceptualize this was to look at like color printers and to explain why we can produce color, all the different colors that we perceive, but using only three actual wavelengths, right? You see that in our computer screens or in printers. Basically, they only use three colors, and yet by adding them together, you can create, all the different colors. Yeah, how does that work then when you have multiple cones absorbing multiple frequencies of light, and then our perception perceives a color from, some complex spectrum of light that we're actually receiving, right?
I guess one way I think about it is that if you're talking about pure light and you're shining the light in somebody's eyes, let's say maybe it's like two pure wavelengths. So let's think about a human. So let's say it's
Okay.
a blue light and a red light. And so that photoreceptor cell is going to weight that light in a certain way. Maybe it's going to have, it's going to give it a higher weight than the other photoreceptor cells in the eye. Conversely, the red is going to be given a higher weight by, say, the red cone pigment. And your brain is able to take those weighted responses and convert that into, our perception of a particular color.
So I had somebody on to talk about hearing, and basically when we hear something, we're getting the whole spectrum of frequencies all at once, and we're perceiving all of them. So our eyes receive a light, and it's filled with the whole spectrum of light. It's weighted differently. So if it's green, it's receiving more light, but it's got all the different frequencies kind of mixed in there, unless you're giving it some pure wavelength, like I guess you do with your experiments. But we don't perceive that whole spectrum. Our eyes kind of bucket it into certain places, right? So it's like if it falls within this frequency, our eyes just grab it and say it's here. But then we kind of use all three of them to determine where on that spectrum it is, depending on how much stimulation each one gets in each of those buckets. And then we can basically fake it then by stimulating just the buckets with our printers. That's kind of how I thought of it.
Yeah, absolutely.
Okay. Yeah, it's kind of weird to think about that you can just basically fool us, give a completely different spectrum than what the thing actually would be in real life, but our eye still just perceives it in the same way.